Good morning, everyone. Today, I'm excited to share our work on mathematical modeling and analysis of multicellular dynamics in morphogenesis, where we explore how mathematical frameworks can deepen our understanding of the intricate processes driving tissue development and organization. First of all, I'd like to introduce you to the fascinating process of morphogenesis. Morphogenesis is the biological process where a group of cells autonomously generate forces and transform their collective structure into a specific shape as an organized tissue. Morphogenesis raises several intriguing questions. How do individual cells contribute to shaping the tissue? And how can these local cellular activities coordinate to control the global structure formation of the tissue? To address these questions, let's start by exploring two key cellular mechanisms that contribute to morphogenesis: apical constriction and cell proliferation. Apical constriction is a process where the apical side of cells contracts, causing the tissue to bend, which is critical for shaping structures like the tissue invagination. Cell proliferation involves cells dividing to increase their number. This expansion contributes to tissue growth and also influences the overall shape of the tissue. Together, these mechanical effects of cellular activities are fundamental to morphogenesis. First, I like to introduce the role of apical constriction during tissue folding. If we decompose the process of the tissue deformation into fundamental mode of deformations, one key mode is folding. Epithelium folding transforms a 2D cell sheet into the 3D. Basically, the 3D structure of epithelium tissue is established by repeating process of epithelium folding. But how does the epithelium folding occur by cell activity such as apical constriction? There is a spatial pattern of contracting cells in the folding which corresponds to a crease line of origami. For example, in the ventral furrow, you can observe a line pattern of contraction. During gastrulation, cells contract in a disc pattern, whereas in tracheal placodes they form a ring pattern. These spatial arrangements of contracting cells are not random. They are highly organized to generate specific 3D structures from flat epithelial sheets. The question is, how does this patterned contraction translate into tissue folding, and what types of 3D structures can be derived from these patterns? So the purpose in this study is very simple. If we have a particular spatial pattern of contracting cells, what is shaped to be folded based on the pattern? For instance, this is a cross-section of tissue along apicobasal axis. If some cells are contracted by apical constrictions, and if compression forces exert on edges like this tissue can bend as invagination. It is a trivial example. However, if we have these patterns in two-dimensional sheet, what kind of three-dimensional shape can emerge from such patterns? To address this, we employ computational simulations to set spatial pattern freely and investigate what is the role of spatial pattern of contracting cells in epithelial folding. To simulate epithelial folding, we employ a 3D vertex model. In this model, the tissue is represented as an aggregate of cells, where the shape of each cell is described as a polyhedron composed of vertices and edges. These vertices and edges are shared with neighboring polyhedrons, ensuring connectivity within the tissue. To capture the dynamics of the multicellular aggregate, the motion of each vertex is governed by an overdamped equation of motion. The left-hand side represents the frictional force acting on vertex I in the local inertial frame. The right-hand side represents a conservative force derived from the energy function U. The energy function U characterizes the mechanical properties of the cells and morphogenic processes at the cellular scale, such as cell growth and apical constriction. In addition to simulating mechanical behavior, the model incorporates cell division through polyhedron splitting and cell rearrangement through network reconnection. We show two major results. At first, we confirm that cell proliferation generates compressive forces and destabilizes a flat sheet shape leading to the sheet buckling, and apical constriction determines the position of the sheet buckling. Secondly, we show two simulation results based on two different spatial patterns. Two spatial patterns are apparently similar to each other but topologically different. The difference in the topology induces the reversing the direction of out-of-plane deformation from each other. Contracting cells are placed in a circular disc which is defined by this inequality. All cells proliferate according to cell cycle. The surface in the field of view is the apical surface. In the movie A, only apical constriction is included, resulting in a slight bending deformation toward the basal side. The deformation magnitude is small. In the movie B, only cell proliferation is included. Because cell proliferation generates compressive stress in the tissue, the flat shape of the tissue becomes unstable and shows a sheet buckling. The shape was very undulated. The magnitude of the deformation is very large. In the movie C, we include both apical constriction and cell proliferation. The tissue showed a large invagination at the constricted region without any other undulation. The elastic energy generated by cell proliferation was used to drive the invagination, with apical constriction fixing the point of sheet buckling instability to only the constricted region. Thus, apical constriction determines the location of the invagination, and cell proliferation amplifies the depth of the invagination. Next, we show how the difference in spatial pattern changes the epithelium folding. We consider two spatial patterns. One is the circular disc, which is shown in previous slide. In this case, the cell proliferation occurs inside the circle only. The other pattern is the circumference of the circle. Here we shall call it circular ring. Cell proliferation occurs in the enclosed region. The area of the cell proliferation is the same in both cases. The two spatial patterns appear similar but are topologically distinct. The circular disc pattern has no holes, whereas the circular ring pattern contains a single hole. Despite this subtle difference, the impact on out-of-plane deformation is significant. From the circular disc, the tissue forms a basally convex shape, while the circular ring generates an apically convex shape. This contrast arises because the epithelial sheet exhibits apico-basal polarity, making the direction of out-of-plane deformation crucial for organ formation. These results demonstrate that apical constriction can deform tissues in both directions, basally or apically. This raises an intriguing question. Why does the circular ring pattern produce an apically convex tissue shape? Focusing on the cellular shape, we can see that apically constricted cell became an apically narrow wedge-like shape in both cases. In the circular disc, wedge-like cells locate next to each other. Thus, the tissue shape became the basally convex. On the other hand, wedge-like cells do not locate next to each other along the radial axis of the ring. At the boundary between contracting and non-contracting cells, the non-contracting cell tilted toward the apical direction to align cell shape to the wedge-like cells. Therefore, from this geometrical restrictions, the tissue shape became the apically convex. Next, we shift our focus to emphasize the role of cell proliferation in epithelial folding. Again, epithelial folding is a fundamental process in development. Examples include the folds in our brain, gyri, the villi in the intestines, and even the wings of insects. These structures are known to emerge in association with cell proliferation. But why do such a variety of folding shapes exist? What determines the specific patterns and directions of these folds? Despite its significance, the mechanisms underlying these complex folding processes are still not fully understood. One key factor influencing epithelial folding is the constraint of out-of-plane deformation. These constraints are imposed by surrounding tissue layers, such as other cell layers, and the extracellular matrix (ECM), including collagen. These mechanical interactions play a crucial role in shaping the final phenotype of the epithelial folding structure. To investigate the role of suppression of out-of-plane deformation, we performed simulations of growing epithelial sheets. In case A, epithelial sheet grows by cell division without out-of-plane constraint, and we obtain a coarse folding structure with a small number of wrinkles. In case B, growing epithelial sheet undergoes an out-of-plane constraint and multiple folds formed in the epithelial sheet. To understand the relationship between wavelength and mechanical property of tissue, we consider tissue shape at energy minimum state theoretically. Considering out-of-plane confinement and bending energies of the tissue, we found that these energies compete with each other, so that the ratio of constraint strength to the bending stiffness is unique parameter to determine the wave number. The wave number is expressed by the power of the ratio of power exponent 0.25. Simulation result shows the power law behavior in wrinkling formation with respect to the strength of restraint. Thus, the constraint is important to determine the wave number of folding structure. In addition, the direction of cell division affects the shape of epithelial folding. This slide shows 3 cases of cell division orientation. When cells divide along their long axis, the folding pattern becomes irregular, like a labyrinth. When all cells divide in the unidirection, the folding pattern becomes parallel lines. When cells divide outward in a radial direction, the folds form circular patterns, like rings. These examples show how the orientation of cell division creates different shapes, which is very important for tissue development. Next, we highlight that the asymmetry in environmental constraints also plays a crucial role in determining folding patterns. As shown in this schematic, the epithelial tissue is surrounded by materials with different mechanical properties, an elastic layer on one side and a viscous layer on the other. This asymmetry influences the direction and shape of the fold, demonstrating how environmental conditions guide morphogenesis. To characterize this asymmetry for understanding its effects on epithelial folding, we define the degree of asymmetry using the parameter lambda, which reflects the relative distance of the basal surface to the elastic material, compared to the total distance between the sheet and both apical and basal constraints. Using the multicellular dynamics model, we investigated how this asymmetry affects the epithelial folding. Now let's see how asymmetry affects folding patterns in our simulations. We have 2 examples here. These tissues grow by cell divisions under different asymmetry conditions. In the first case, where constraints are symmetrical, we see a labyrinth-like pattern forming. In the second case, where the sheet is attached at the bottom, we see a pattern dominated by spot-like protrusions. This shows that even small differences in environmental asymmetry can lead to very different folding morphologies. Let's see how folding patterns change as the degree of asymmetry increases. When lambda is 0, we observe a distinct pattern of protrusions. As we gradually increase lambda, moving toward a more symmetrical constraint, the pattern begins to transition, becoming less distinct and more labyrinth-like. At lambda equals 0.5, where the constraints are perfectly symmetrical, we see a clear labyrinthine pattern. As lambda increases beyond this point, the pattern shifts again, forming an array of invaginations directed toward the basal side. If you are familiar with Turing patterns, you might notice that this progression is similar to how patterns change with respect to parameters in Turing's equations. This similarity suggests that the folding mechanism may share underlying principles with Turing pattern formation. But this is not the Turing equation. This is the Swift-Hohenberg equation. To gain a deeper understanding of the pattern formation mechanism, we introduce an energy functional that governs the system. In this equation, the first term represents the bending energy, the second term accounts for the axial strain energy, and the last 2 terms describe the constraints imposed by the external environment. The system evolves to minimize this energy. From the energy functional, we can derive a governing equation for the dynamics of the sheet deformation as known as the Swift-Hohenberg equation. Here, you see that the degree of asymmetry is the key parameter for pattern selection. This shows how a simple change in environmental asymmetry can lead to dramatic shifts in epithelial morphologies. Now look at 2 examples. First, the finger-like villi in the small intestine. Their development involves epithelial folding, with the basal side anchored to the extracellular matrix. This anchoring creates an asymmetry in the constraints. In vivo shapes of villi are observed as finger and leaf-like structure, while simulations successfully reproduce the same variety of shapes even when lambda equals 0. Now let's turn to the complex folds of the beetle horn primordium. As you can see in image, the primordium develops within a confined environment, but with spaces on either side of the epithelium, suggesting a more symmetrical constraint profile. Let's look at the beetle horn primordium. Its surface has unique wrinkles forming continuous ridges. Interestingly, when we set our model to symmetrical conditions, we see a similar labyrinth pattern. This close match between nature and our model provides insights into the formation of these biological structures. So far, the multicellular dynamics model has been used to clarify the mechanical roles of cells under simplified physical conditions, rather than analyzing real experimental systems. But can this model help us understand actual biological processes? Can it extract meaningful information from real experimental data? This is the question we aim to address. Here, we focus on neural tube formation in Xenopus, an example of epithelial morphogenesis. This is the schematic of process of neural tube formation. Xenopus neuroectoderm is stratified as double layer. At first, a central region of neural plate at the superficial layer is thickening by cell elongation. Then, a bending of the neural plate is occurred due to apical constriction of the cells, as we saw in the previous slides. Thirdly, superficial layer cells are pulled by cell migration in a deep layer. 3 cell events, cell elongation, apical constriction, and cell migration, are supposed to be important for neural tube formation. So the hypothesis to be examined here is that 3 cell events is mechanically sufficient to drive neural tube formation. Using the model, we can test whether a phenomenon can be explained solely by the working hypotheses derived from experiments. So we express each cell event in terms of energy function. Cell elongation and apical constriction are expressed by elastic energies with respect to cell height and apical circumferential ring, respectively. Cell migration is expressed by a potential energy of non-neural migrating cells. Here FCM is migrating force, and XJC is position of migrating cell from the midline. In the model, we take into account the double-layer structure of neuroectoderm bounded by non-neuroectoderm. Then we express 3 cell events using energy function U, and activated these energy function based on onset time of the event observed by the experiment. This is the simulation result. Here, as shown, the neural tube formation is achieved through these 3 cell events. To compare the simulation result with experiment, we quantified cell and tissue shapes in both simulation and experiment at several stages, and found that the simulation of the control model coincided with experiment very nicely. We inhibited cell events in silico and investigated the final shape of the tissue. This is the control as shown in the previous slide. In the case of inhibition of both cell elongation and migration, we found that neural plate bending was achieved through apical constriction, but the magnitude of deformation was very slightly. It's obviously insufficient to the neural tube formation. In the case of inhibition of cell migration, the bending of the tissue was enlarged comparing with inhibition a case, but the neural tube closure was not accomplished. Therefore, all 3 events, apical constriction, cell elongation, and cell migration, are necessary for successful neural tube formation. We virtually altered the mechanical properties of the underlying sheet at the basal side, ranging from soft to hard. These images show the final shapes of the neural tube at closure. When cell elongation was inhibited, the final shapes became highly sensitive to the elasticity of the underlying matrix. This indicates that without cell elongation, neural tube formation is more affected by developmental noise from the mechanical environment. In contrast, in the control model, the neural tube maintained a consistent shape regardless of the sheet elasticity. This suggests that cell elongation plays a role in protecting tube formation from mechanical influences, ensuring the robustness of the final shape of the neural tube in Xenopus. We propose that this robustness may be the physical significance of cell elongation. Thus, using the model, we can test whether phenomena can be explained solely by hypotheses derived from experiments and uncover their physical meaning. We see how experimental observations lead to hypothesis formulation and testing. Hypotheses are formulated based on experimental data. These hypotheses are then tested through simulations. For example, a hypothesis might predict how a flat tissue transitions into a 3-dimensional structure, as in gastrulation or neural tube formation. Simulations provide a powerful tool to test whether these hypotheses align with experimental observations. This approach is effective for verifying known hypotheses. However, without a working hypothesis, constructing any multicellular model is impossible. Therefore, we suggest a new method to infer causality in morphogenesis. Once the causality is inferred using this method, it can be tested as a working hypothesis through both experiments and simulations. Again, we shall consider a sheet-like tissue which has curved surface morphology. Many curved biological shapes are formed from the growth of cell sheets. In their early stages, these tissues are nearly flat and simple, but later, they develop into complex curved shapes. A key factor driving this transformation is differential growth, a growth mode characterized by spatially biased growth rate. To help you understand the key point of this topic, let's try 3 quizzes. In these quizzes you will see a final shape and think about its cause. Here, the cause means the spatial distribution of tissue growth, or in geometry, the area expansion rate distribution. The non-uniform distribution of expansion rates is very important for making 3D shapes. For example, look at this protrusion-like shape. What kind of area expansion rate distribution do you think creates this shape? The answer is a centralized area expansion pattern where the central region grows at a much higher rate. When the red area expands significantly, it causes the tissue to form this protrusion-like shape. Now let's consider how this frill-like shape is formed. Can you guess the distribution of expansion? In contrast to the previous example, the frill-like shape is formed when expansion rates are concentrated at the periphery. For the final question, let's think about which distribution might create this clover-like shape. Do we have anyone who think it's pattern 1? How about pattern 2? And finally, anyone for pattern 3? The answer is pattern 1, the ring-like expansion pattern. Surprisingly, even complex shapes like this clover-like structure can result from relatively simple expansion patterns. However, reverse engineering these patterns from a 3D shape is very challenging. This is the key point I wanted to share today. To solve this, we are exploring mathematical methods to address this inverse problem. Our research aims to develop a method for inferring the area expansion rate distribution during the growth process of complex, three-dimensional morphogenesis. To solve this inverse problem, we make an assumption about the growth process. It takes place in a locally isotropic manner. In mathematical terms, the shapes before and after growth are connected by a conformal map, and our goal is to construct this conformal map. The method for constructing a conformal map onto a planar domain is well established. First, we perform conformal planarization for pre- and post-growth shapes respectively. And then we take the image of the post-growth shape onto the pre-growth shape using the inverse of the pre-growth shape's conformal planarization. In this way, we can construct conformal map between pre- and post-growth shapes as composition of fc_post and inverse of fc_pre. Once we construct the conformal map, we can compute the area expansion rate at each triangle as the area A post over the area A pre. We validated this method using various artificially generated shapes. These shapes in the top row were created by applying the area expansion rate distributions shown in the middle row, which are the ground truth distributions. The bottom row shows the results of our inference, and as you can see, they closely match the ground truth distributions. This demonstrates that our method successfully inferred the correct area expansion rate distributions. Moving on to the practical application for the formation of rhinoceros beetle horns. Beetle horns develop inside the larval head by growing epithelial sheets through cell proliferation. In the final step, they simply unfold the wrinkles without any change in area, so we do not consider the pupation process. In the beetle horn formation, the most significant 3D shaping happens during the mushroom-like shape formation, so we applied our method to this process. For this growth process, our inference method produced this distribution. It suggests that localized concentrated areas of expansion are crucial for the formation of the mushroom shape. Furthermore, there are regions of particularly high expansion rates at the left and right edges of the expansion region. To verify that this area expansion rate distribution can indeed form the mushroom shape, we performed a forward growth simulation. This simulation confirmed that this expansion rate distribution could explain mushroom shape. Additionally, experimental observations reveal a higher frequency of cell division at the left and right edges of the horn primordium. Our inference results also indicate a notably higher area expansion rate at these same edges. This correspondence between experimental observations and our inference demonstrates the consistency and validity of our method. We have developed an inference method which helps explain the causal relationship between differential growth and tissue shape. We believe that this method is applicable for various morphogenic processes. To summarize, this framework connects experimental observations, hypothesis formulation, simulations, and inference computations to understand biological morphogenesis. Based on experimental observations, we create hypotheses, such as spatial patterns, temporal histories, or the effects of surrounding structures. These hypotheses are tested using multicellular dynamics simulations, which predict and analyze 3D tissue deformation from cellular mechanics. When we don't have clear hypotheses, inference computations help us. This method uses shape information to infer causality. We can then test this causality through experiments and simulations. This process creates a feedback loop that improves our understanding and guides future experiments. By combining experiments, simulations, and inference, this framework helps us find new causal mechanisms and better understand the principles of morphogenesis. Thank you for your attention. This slide summarizes the key references and acknowledges the support received for our research. These works were supported by Japan Next Kakanhi grants. I would also like to express my sincere gratitude to all collaborators for their valuable contributions. If you have any questions or would like to discuss further, please feel free to contact me.